And now what we've all been waiting for, inheritance of single traits, part 3, the third and final part. Yay! All right, moving on. So, finally, the most funnest part of genetics, the Punnett Square. Okay, so a Punnett Square is a prediction of genotypes and phenotypes of offspring of a particular cross, okay? And it is just that. It is a prediction. This is saying, if you make this cross, this is what could happen. It is not saying this is what does happen. So make sure you keep that, separate. It is a prediction. So Punnett Square predicts the offspring's genotypes and phenotypes from different combinations of parental gametes. Okay? So in order to do a Punnett Square, we got to take a couple steps here. First, we're going to assign letters to the alleles. Okay? Uppercase for dominant and lowercase for recessive. Okay? Then we're going to determine the gametes from both parents. And then we write the gametes from each parent. We won't worry about this fractional proportion thing yet, but we're going to write the gametes from each parent along each side of a 2x2 grid, like so. 2x2. Okay? And then you fill in the genotypes in the box. Okay? So we can put, so say I have the following cross. We'll do our true breeding homozygous dominant versus homozygous recessive cross of pea plants with purple flowers and white flowers. So if this is my parent, it's can produce the following gametes with a dominant allele. That's all. And the recessive parent can only produce recessive allele gametes. So we put the gametes on either side. So the dominant parent can make dominant gametes. The recessive parent can make recessive gametes. And then in the boxes, we're going to make our zygotes. If we add this and this, we get to put those two gametes together. We get that as a zygote, heterozygous. This will go here with that. Same thing. So we're combining the gametes in the boxes to make our zygotes. And that's where the zygous in heterozygous or homozygous comes up. So then we have the genotypes within the boxes. And this genotype has one allele of each. So it would be heterozygous. A zygote with one of each allele. Okay? And we're going to, we'll talk about the genotypes and phenotypes in a second. But we are going to talk about a genotypic ratio and a phenotypic ratio. Okay, so the ratio compares the different genotypes, and the phenotypic ratio compares the different phenotypes. So let's see how that's going to work. We can, again, and this is my phenotypic ratio. Okay, so we're going to always take dominant and recessive rules into account. Okay, so here we have a sample Punnett Square. Okay, and if we have a cross here, we have a heterozygous parent that's going to be self-fertilized. So the alleles that get put into the gametes, okay, is one dominant allele and a recessive allele in each of these gametes, and a dominant allele and a recessive allele in each of these gametes. Then when we fill in our cross, our Punnett Square, You bring down those gametes, and this box gets homozygous dominant. This box gets one of each heterozygous. This box is also heterozygous, and this box is homozygous recessive. Okay? So if you look, 1 out of the 4 boxes is homozygous dominant. 1 out of the 4 boxes is homozygous recessive. And 2 out of the 4 boxes is heterozygous. So my genotypic ratio over here is 1 to 2 to 1. 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Okay, but then if we look at our phenotypes, the physical expression, I have 1, 2, 3 flowers that are purple and 1 flower that is white. So 3 out of the 4 are purple and 1 out of the 4 is white. So my phenotypic ratio is 3 to 1. Okay, and I always want the ratios written like 1 to 2 to 1 or 3 to 1, not like 25, 50, 25%, not 1/4, 1/2, 1/4. I want them written as ratios. Okay. So that's our Punnett square, again, for a single trait, because we're still looking at inheritance of single traits. Okay. And again, actually, before we move on, remember this is a prediction. Okay. It is not something that is actually going to happen. This is the odds that this situation, that any of these situations happen. All right, so let's talk about a test cross, which is a special type of single trait crosses. Okay, and test crosses are used to determine whether an organism with a dominant phenotype is homozygous or heterozygous. Okay, because if I have these 2 genotypes, they're both the same phenotype. They're both purple. So if I wanted to know if my organism was homozygous or heterozygous, I can do what's called a test cross. Okay? So a test cross crosses the unknown dominant phenotype organism with a homozygous recessive organism. Okay? If the dominant phenotype organism is homozygous, only dominant offspring will be produced. You'll only see the dominant phenotype. If the dominant phenotype is heterozygous, then approximately half of the offspring will be the recessive phenotype. Okay, and let's just, I think there's more room on the next slide. Oh, and we have a test cross. Okay, so again, we don't know, we have a purple flower. We don't know if it's homozygous dominant or heterozygous for the genotype, and we want to find out. So if it's homozygous dominant, okay, the gametes can only have one possible allele. It's all going to be a dominant allele. From our homozygous recessive, they can also only give one allele to the gamete, so it'll only have a recessive allele. So all offspring would be heterozygous, and you would have all purple. So all the dominant phenotype. If we have a heterozygote, there's 2 different types of alleles that can be put in the gametes. So you can have some gametes that have a dominant allele and some gametes that have a recessive allele. Okay, and that means, let me creep this up here a little bit, that if we cross it with our homozygous recessive, okay, we can get heterozygotes and homozygous recessive. Okay, so we will see the recessive phenotype. So if the parent is heterozygous, then you will see the recessive. If the parent is homozygous, you don't see the recessive. You get all dominant phenotypes. Okay, so that's the little. We're going to do tons more genetics problems in class with using Punnett squares, but that's like a little introduction to the world of Punnett squares and how they work, genotypic ratios, phenotypic ratios. So that should give you a decent base for the work we're going to do in class. Okay, the end.